Welcome. In this class, I will introduce you to what database is basically about in bioinformatics. What is database? What do we understand by database? What are the essence and significance of database in bioinformatics study? But this time around, we will like to I would like us to see into the meaning of database on a general term. A database is a structured collection of records stored in a computer system. So we have different kinds of database. We have financial database, we have computer database, we have a lot of database. But this time around, we'll be looking at biological database. We'll be focusing on biological database in this study. Now, what are biological database? What do you understand by biological database? Now, a biological database are libraries of life sciences information collected from scientific experiments, published literatures, high throughput experiment technology, and computational analysis. So every information that comes from science experiments, published literatures on science, on biological content, and then high throughput experimental technology and computational analysis are being done. So now this time around we are looking at computational biology. So the aspect of the computational analysis actually deals with bioinformatics in biological studies. So let's look at this. Now, they contain information from research area including genomics. Now these are database. Now they contain information from research areas including genomics. And if we're talking about genomics, we're talking about DNA. We're talking about DNA data. We're talking about DNA sequences. Now we also talk about transcriptomics. Now transcriptomics are RNA, RNA sequences, RNA analysis. So now we also talk about proteomics. We're talking about proteomics. We're talking about proteins, microarray gene expressions, and mutations. Looking for hidden information. What certain gene coded for in each of those data, in each of these sequences. So that's gene expression analysis. And phylogenetics, looking at evolutionary relationship, similarities and differences, and a lot of things. So information contained in biological database include gene structure, gene function, gene structure, clinical effects of mutation, as well as similarities and differences of biological sequences and structures. So many databases also provide bioinformatics tools such as BLAST for finding specific sequences or annotations. There are hundreds of genomic databases. There are hundreds of them. And we also talk about the tool, which is BLAST. We'll also talk about it once we get to the internet aspect of this introduction. So, but first let's carefully study some of them one after the other. So, examples of genomic database. We have primary DNA sequence database. Now, this particular database actually stored DNA sequences. DNA data, molecular data that are specifically DNA. So whenever you are looking for a DNA sequence or a DNA data, you can actually retrieve it or kind of download it from these particular databases that we're going to be dealing with. But there is something that is very specific and peculiar with this database. It is that every data that are being stored on these databases, they can be controlled by the submitter. So you can be a submitter. Anybody can be a submitter. I can be a submitter. So I can just submit the data or whatever I'm working on the internet, on the database. So anybody can retrieve it. But then I have a full control to
control, to maybe update, to delete, to edit the data that actually stored on the database. So I have the access. Now let's look at those database. We have EMPL, nucleotide sequence database. Yeah, this is a database actually, and the essence of this database is that it stored nucleotide sequence. So it stored DNA nucleotide sequence and RNA nucleotide sequence, but it doesn't store protein. So that's the specificity of this particular database. So you can actually browse this particular database on Google just to see, maybe to go into the website, to get the website address, and then just go through it and see how you can download. But then we'll work on the practical applications of maybe downloading, retrieving, and doing a lot of things on it. Now we'll be using NCBI GenBank. We'll be using NCBI GenBank. We'll be using it to do a lot of things. So I'll be talking more about NCBI GenBank in a few seconds now. So we have DDBJ, that is DNA Data Bank of Japan. So Japan owns this database. Yes, he owns it. Now we have INLSDC, we have International Nucleotide Sequence Database Collaboration. So this is also another database. Now, there's something that is very significant. There's something called data flow. Now, the data that is being stored in EMPL here can actually be synchronized into NCBI. Whatever that is found in NCBI can also be synchronized to EBI and DTBG. So just like that, the data in each of these databases have been synchronized round. So you can actually browse a particular database on NCBI, whereas it's actually gotten from DTBG or stored originally on DTBG. So that's something about that. It's very significant. Now we have primary protein sequence database. Now, say this primary protein sequence database actually is similar to primary DNA sequence database. Just that the difference is that the database here is only stored protein. So you can only find protein on this database, but you can't find DNA on the other primary DNA sequence database. So some of the database here are SwissProt, PRR, and Tremble. So you can just browse them on Google and you get them. So maybe you want to retrieve a protein sequence. You want to work on protein sequence, protein data. So you can just go to this database and then retrieve them, search for them, and then download them. So we have UniProt. So UniProt is used in a universal way. So most people use UniProt. So to search and download protein sequence data. Then there's something that is very peculiar about this database too. So we have the protein knowledge base consists of two sections. Like this protein knowledge base consists of two sections. So one is the SwissProt, and the essence of this is that they manually annotate and review. So you can manually annotate some of the sequences on this UniProt, and then we have Tremble automatically annotates, not reviewed. So it's around the functions and features of this database. We have Interpro database assay. So this actually also work on protein sequences. As you can see, Interpro different protein families, domains, region, repeats, and sites according to matches against member database. So these are the functions of Interpro. So, but the basic essence of this Interpro is that you can actually, search and download the sequence data that is only peculiar to protein. Peculiar to protein. So we have genome database. So this database actually stored genomes of organisms, genome of plants, genomes of animal. Maybe you want to, you are looking for a particular genome. Maybe for E. coli, you want to download the entire genome of E. coli. So you can actually go to NCBM database. You can go to UCSC Genome Bioinformatics. So you can go to eSemble. So a lot of them like that. So actually, we have come to the end of this lesson, introduction to database. So in the next lesson, we'll be going to introduction to internet tools.